All right, in this section we take literally everything that we just put together and we tear it all apart. Okay, so our essential question for this one is going to be, how do we describe traits that don't fit in Mendel's original, kind of, we'll say don't fit within the original Mendel's laws, because he was the original. He's the first dude. So how do we describe things that aren't going to fit within that category? Well, there's going to be a lot of them. So let's start here. So Mendel's P crosses always look like one of the two parental varieties. That's what we call complete dominance. So one particular trait is completely dominant over the other one. If the other one, if the dominant one is there, we don't even see the, recessive one. Okay, but some characteristics, the appearance of the F1 hybrids falls between the phenotypes of the two parental varieties. This is called incomplete dominance. And so incomplete dominance is where we have a blend. Kind of our heterozygotes are going to look like a blend of the two parents. Okay, so let's say we have snapdragons. This is our kind of major thing here is snapdragon flowers. Snapdragons. Okay, so we have red snapdragons and we have white snapdragons. Well, when we cross those, we get pink snapdragons. So they're kind of a blend of the two. If you cross our pink snapdragons together, you end up with one of the red, two of the pink, one of the white. So you end up with the same thing that you have before. It works the same way with the heterozygotes, but instead of us having three the same phenotypically, we'll have, or instead of having two phenotypes appear, we'll have three phenotypes appear. See if I can get my words out today. So one example of incomplete dominance in humans is called hypercholesterolemia. You're welcome. Okay, so this is where we have dangerously high levels of cholesterol, and they occur within the blood. Heterozygotes have intermediate high cholesterol levels. So sort of what it would look like, just to give you an example, normal cell has these kind of LDL receptors, low density lipoproteins receptors. If you have a severe disease that you don't have any of them, so the homozygous recessive is that way. But if you have mild disease, you're heterozygous and you'll have just fewer. So it sort of looks like a blend between normal and severe. It's just somewhere in the middle. Okay, although each individual carries at most two different alleles, we can only do two at most because of the whole diploid or diploid thing for a particular gene. In cases of multiple alleles, there are two possible alleles that exist. Okay, there can be more than two possible alleles that exist in a population. I'm going to get my words out eventually. So a really good example of this one is the ABO blood group. Okay, so phenotypes actually involve three separate alleles for a single gene. So we have A as a type, we have B as another allele, and we have kind of O as an allele. So the four human blood groups A, B, and O result from combination of these alleles. A and B alleles are expressed in heterozygous individuals, making both codominant. So codominant isn't really like an incomplete dominance. They kind of make this blend. In codominance, it's kind of like it's a patchy type thing. Okay, so what happens is we have the A blood type, and it will express A here. This is a whole interesting thing. For B, it'll represent Bs, like with these little kind of, the antigens, they look different, but we've represented them as little spheres here. If we have AB, notice it's kind of patchy. There are triangles and there are spheres. It shows both of them. If you have O, then you have neither. So I happen to be O, so my particular genotype is little I, little I. This whole I situation, big I with a little A over it, it actually represents the I is trying to tell us a locus or a location. So this is the same way we kind of look at that top letter. Oh, I can't circle it, I'm sorry, but big, if we have big A, then it's still both of the dominant. If we have big A, little I, it says we have the recessive. So that's kind of a weird one. We'll have to talk about blood a little bit later, but this is kind of a fascinating one. So if you have A, your body can get blood from O donors or A donors, not B or A and B. If you have B blood type, you can receive blood from O or from B, but not A and not A and B. If you're AB, you can receive blood from anybody. So we call these kind of the universal acceptor. So if you hear those terms. Okay. If you're O, you can really only receive blood from O. Okay. But if you look at this, everybody can take O, so this is why we call this the universal donor. So we can talk about that one a little bit more about how that works, but that gives you a little idea of what we're talking about. Actually, when we go through all kinds of practice problems, we'll talk about it. We have a thing called pleiotropy that occurs when one gene influences multiple characteristics. We'll look at some examples. Okay. Sickle cell, for example, is pleiotropy. So the disease affects the type of hemoglobin produced and the shape of the red blood cells, and it causes anemia and organ damage. So sickle cell and non-sickle cells are codominant, so it's kind of a patchy thing. It appears both. Carriers of sickle cell disease have increased resistance to malaria. This is one of the reasons that evolutionarily sickle cell persists, specifically in Africa and malaria-carrying countries with, relatively little health care, is because it actually helps to fight off malaria. So you can see there's kind of a patchy appearance that if you're the heterozygous, so for sickle cell, like if you have sickle cell but you carry sickle cell, then some of the cells will be normal and nice and round, and some of the cells will actually be sickle shaped. So it's kind of a half and half. It's a patchy type of appearance. So an individual homozygous for sickle cell produces sickled or abnormal hemoglobin. That also affects the shape of the cell. That gives organ damage. It has other effects like joint problems, weakness, anemia. So this is pleiotropy, where we have one particular trait that affects so many other things. Okay, sometimes we have what's called polygenic inheritance. So poly, again, is many. Genic is just like it sounds, it's many genes. So many genes can affect a character. So this is which a single phenotypic character results from the additive effects of two or more genes. So many genes affect one type of character. Your skin color is an example of polygenic inheritance. Okay, so you have a range. There's actually, like this one showing us about 3 different genes that are working here, and the way that we put all these things together is in this ginormous Punnett square, and it gives us a whole range of skin colors. So this is polygenic. When we have a range, like skin color, or you could think height, when there's a range, it's usually polygenic. Okay, just to give you, like, the height is a bell curve, so it's polygenic as well. Many characters result from a combination of heredity and environment, so you have to kind of include both. So skin color is affected by exposure to sunlight. Heart disease and cancer are influenced by genes and the environment. So if you go out in the sun all the time, you might have nice dark skin, but maybe it gets, for some of us, it probably get leathery one day. Okay, but heart disease and cancer have genes that kind of influence those. Identical twins show that a person's traits are the results of genetics and their environment. Okay. And so here's kind of, again, it's a range of different skin colors that we can see here. So that tells us that this is kind of a polygenic trait. Okay, here's two identical twins, and they've got some environmental factors and how they look a little different as well. Okay, perfect. Chromosomal basis of inheritance. We're at 10 minutes. This is a great place to take a break, chunk of notes, come on back. Oop, went too far. So the chromosome theory of inheritance states that genes occupy specific loci or positions or locations on a chromosome. So chromosomes undergo segregation and independent assortment during meiosis. Okay, so we do those. Mendel's laws correlate with chromosome separation and meiosis. The law of segregation says that pairs of alleles separate from each other during gamete formation via meiosis, so alleles separate. And it depends on separation of homologous chromosomes in anaphase 1. The law of independent assortment states that each pair of alleles assorts independently, or other pairs of alleles during gamete formation, so they're not dependent on another allele to tell them which way to go. And this depends on the alternate orientations of chromosomes in metaphase 1. So alternative orientations. Okay, so we could look at them. Here's our two possible arrangements. And then they go a little bit further, so that gives us all kinds of different varieties for gamete formation, particularly with fertilization and all that kind of stuff. Okay, and we could do this with a lovely dihybrid cross, which we will practice a bit later. So Bateson and Punnett studied plants that did not show a 9:3 to 3:1 ratio in the F2 generation. And what they found was that we have things called linked genes. So genes that are located close to together, and they happen to be on the same chromosome. Well, those genes will have to go together into the gamete because they sit on the same chromosome. So they do tend to be inherited together. So for every genetic rule, we have a better rule that breaks it. So if we say P and L are here on those chromosomes, there's a good tendency that we'll keep the big P, big L together, and they'll keep the little p, little l together. So these guys, when they do the whole thing for kind of long pollen and stuff like that, we're figuring out that we're getting something pretty close to just a regular, 3:1 ratio, despite the fact that we've got, two genes at work here. So this is not a 9:3 to 3:1 ratio. Okay, we can talk about that one. So crossing over between homologous chromosomes produces new combinations. So those can potentially link or separate linked genes. Okay, and those would be recombinant gametes. So the percentage of recombinant offspring is the recombinant frequency. So how that works is that, if they're a little bit further apart, then we can get some recombinant gametes. So if you notice kind of here, they'll recombine because they're far enough apart that crossing over occurs and switches them up a little bit. So our parental gametes and our recombinants don't look the same. So Drosophila, because Drosophila. So they experimented with the gray body and long wings and actually found that there tends to be recombinant frequency was only 17%. So that's not a whole lot. We kind of expect like 50% to happen. But because it's 17%, we're kind of assuming those guys are closer together. So when examining recombinant frequency, Sturtevant, one of Morgan's students, found that the greater distance, so this is the main point, the greater distance between two genes on a chromosome, the more points there are between them where crossing over can occur. So recombination frequencies can thus be kind of used to map relative positions. We call that a linkage map. Okay, so I'll show you kind of what that looks like. So if we say 17% overall for these particular genes, if we've got recombinant frequencies in between, then we can sort of get an idea about how close or how far away it is, because the lower we have frequencies, the lower percentage of recombination, the closer they are. closer together, we'll say lower recombinant percent frequency. Something like that. Oh, that came out as a G, even though it shouldn't have. Okay. So this is actually how they're able to kind of tell like red eyes and gray body. So red eyes, gray body tend to be pretty close to each other and have a smaller kind of percentage of recombination than, let's say, let's say maybe brown eyes and short iristi, which are like its little antennae. So those guys tend to have higher recombinant frequencies because they're further apart. Okay, for Drosophila. Okay, now we can look at sex chromosomes and sex-linked genes. So many animals have a pair of sex chromosomes designated as X and Y. That's how we know, at least for humans. So among humans and other animals, if we have one X and one Y, then it's a male. XX are females. And in addition, human males and females both have 44 autosomes, non-sex chromosomes that are essentially the same between the two of them. In mammals, including humans, the Y chromosome has a crucial gene, the SRY, for the development of testes. With the basic thing, what it does. So the absence of the SRY gene directs ovaries to develop. That's about it. Though I will say other research does suggest that there should be maybe more to it than that, but that's what we know. So X is kind of regular sized chromosome, and little Y is a little tiny dude. Okay, so males and females, we have 44. That's our diploid number plus the X and Y, so that would be 46. When we get to our gametes, we can have 22 and an X or 22 and a Y. For males, females can only contribute 22 and an X. Okay, so when those recombine, really it's the male, that's kind of, it's his genetics that determine whether it's female or male. So all that historical stuff about behead her, she did not bear me a son. It's like, well, that was your fault, buddy. But there are some other systems just to make note of. You don't have to know these, but I just think it's interesting to share with you. So grasshoppers, roaches, and some other insects have an XO system. So the O is an absence of a sex chromosome. So females are XX, males are just X. Okay. And certain fishes, butterflies, and birds, they have this ZW system. So males are ZZ, females are ZW. So the heterozygous here is the female in this case. Oop, I went too far. Some organisms lack chromosomes altogether. Most ants and bees, sex is determined by chromosome number, which is really fascinating. So females develop from fertilized eggs and are diploid. Males develop from unfertilized eggs and are fatherless and haploid. That's why they call them drones. It's because they just, it's that kind of thing. So the XO system, if we have males with just one X, females with two X's, ZW system, males is ZZ, females is ZW. Chromosome number 16 for males, females have 32 chromosomes. So there's some other things going on. In some animals, environmental temperature determines sex. So for some species of reptiles, the temperature at which the eggs are incubated during a specific period of time determines whether the embryo will be male or female. So you do have some environmental things that work on that one. Global climate change may therefore impact the sex ratio of many species because if we have warmer temperatures, then maybe we just develop a whole bunch of females. So we also have some sex-linked genes, and they're located on either of the sex chromosomes. So an X chromosome carries many genes unrelated to sex. So the inheritance of white eye color in the fruit fly is an X-linked recessive trait. So look kind of what happens when we talk about this. So a female will have to have two copies because it's XX. Male of white eyes has X with a little r. So again, the X is telling us the location, whether it's on X or Y. So if you do kind of the Punnett square over here real quick, Like we have XR, XR, so this is our female. And here's our male. And he's got X with a little r and Y. Well, we've got, I know I'm going to have to write small here. Oops, just deal with it. Little r. Then we have X, R, and Y. X, R, and Y. So none of these will express the trait. That's what they're getting here with, is that they're all going to be R, or all going to be red because of the things that are showing here. So male or female, all the offspring are red-eyed. But now let's see what happens when we have kind of a female who's heterozygous. Let's see if I can make it bigger this time before I mess it all up. You can sort of see, so we've got X, R, and X with a little r, so our female up top and our male here. Okay, so we've got 2 copies. Again, all the females are good because they're representing, that's basically what this is that all our females are coming out fine because they've got the dominant trait there. So the males are a little bit different. We've got X with a big R and Y, so there's 1 copy of the traits who's red-eyed. But because there's this little r here, he doesn't have anything else to mask the expression of this little r. So it comes out wide-eyed. Okay, so that's what kind of affects thing there, is that only 1 copy for males of these X-linked traits actually kind of changes things. So again, if male is this way and female happens to be heterozygous, then we can get a female finally that expresses that trait, but a male also. So really, it's for particular sex-linked traits, like color blindness, it's more likely for a male to get it than a female. Okay, so most sex-linked human disorders are due to recessive alleles, and they're mostly seen in males. So a male receiving a sex-linked single X-linked recessive allele from his mother will have the disorder. A female has to receive the allele from both parents to be affected. All right. Okay, so some examples really quickly are hemophilia. So it's excessive bleeding. They just can't blood clot. Red-green color blindness is another example. And then Duchenne's muscular dystrophy is a condition characterized by progressive weakening of the muscles and a loss of coordination. So that's another potential one. It's pretty not so good. Okay, we can sort of talk about this one. So if we have a kind of hemophilia, we talk about how these kind of pedigrees work. The squares are always male. Sorry, the best way is males are squares. It's the best way to remember it. So he didn't have it, but we can see this X-linked thing, so let's use H. So he had big H and then a Y, and she has big H and then little h. Okay, so when they have a kid, she's a carrier, right? Big H, little h. Lewis is good, he's big H, Y. Alexandra's good, big H, little h. The Tsar Nicholas II of Russia is big H, Y. But then we get to Alexis, the male down here, and he just happens to get this trait. So he is little h and y. So he represents, or kind of he expresses the hemophilia. Okay. So the Y chromosome provides, we'll just go ahead and finish up here. I know you can take a break if you want to, if you need to, but we are very close. Okay, so the X chromosome provides clues about the human male evolution because the Y chromosomes are passed intact from father to son. So mutations in the X chromosomes actually reveal data about the recent shared ancestry, which is really interesting. So just a kind of crazy connection, in 2003, geneticists discovered that about 8% of the males currently living in Central Asia have a Y chromosome of striking genetic similarity. So this further analysis traced the common genetic ancestor to a single man living about 1,000 years ago. So in combination with historical records and the data, led to the speculation that Mongolian ruler Genghis Khan may have been responsible for the spread of the telltale chromosome to nearly 16 million men living today. You can make whatever speculations you'd like to about Mr. Khan, but I will not. I won't say a thing. You can make our own judgments here. Okay. So with that, that is it. So we have finished it up. There is a lot going on in this section, and so next time we're going to practice Punnett squares for both normal Mendelian genetics, and we'll practice Punnett squares for not normal kind of Mendelian genetics, and we'll talk about them and whatever. So, but if you have any questions and these things don't make sense to you, or if it's unclear, shoot me an email and we'll get it figured out, or come by tutoring or flex time and we'll get it together.